So, following the bulk of Silke, I will continue talking about active fluorescence techniques. And in this case, is the cardfish or TSA fish to detect and identify the parasites. The name, they mean the same, exactly the same, different names for the same technique. One means catalyzed reported deposition, and the other one, thermite signal amplification. It's exactly the same, okay? And then why do we want to use this technique, okay? In this case, we have organisms that are difficult to be seen or distinguished by microscopy, like for the other stainings that we saw before. But in this case, we want to target the organism we want to see at the levels that we need, species level, genus, family, whatever, okay? It relies on designing molecular proofs that are target specific. As I said, the specificity is that one that we need, that we want. And for that, we need the molecular information of our organisms and also of the other things around, because we need to validate these proofs to ensure specificity. We want to avoid false negatives and false positives, because if the proof is not specific enough, maybe it will attach to other organisms that are not those that we want to stain or to see, or maybe it's not good enough and we are not covering all the diversity that we want to see in our samples, and we are just staining some of the species or the organism. If we have a proof that has been published and used many times, we will have it has been already validated, but the perfect world doesn't exist. And then we usually have to design the proof, validate it, make all the tries, and it's a really laborious work to ensure that it's working how we need to do. And well, it can be combined as before with other stainings, like stains of DNA, like DAPI, the cell walls, like chalcofluor, or we can also use double hybridization, targeting different taxonomic levels, and we can also use these filters or these samples to observe them under different microscopy techniques, like fluorescence, confocal microscopy. My talk is really short because actually I prepared a protocol that we will share with you today or tomorrow, and then you will see all the protocol, all the different steps, everything really details with all the solutions that are needed, how to prepare everything, and then you can have a look, and if you have any question, any doubt, you can ask me directly, okay? But I will just give a general overview of how it works, okay? We have our sample. We fix it. We usually fix it with formaldehyde. It works well for the fixation. Another fixative like glutaraldehyde, sometimes they produce some background noise, and we want to avoid that. So formaldehyde is working perfectly for us. We filter, and then we have these filters that we dip them in agarose just to ensure that we don't lose our sample, because in this technique we have a lot of washing, okay, and sometimes you could lose your cells. And then we cover the membrane with agarose, and then at that step there's a permeabilization and inactivation of peroxidase, okay? Depending on your target organism, you need to permeabilize your cells or not. Okay, for eukaryotes, you don't do that. For bacteria, you need to permeabilize the cell wall of the bacteria, because you want that the proof goes inside the cell. For the fungi, you need to do the permeabilization because the ketone wall also has to be permeabilized, okay? And then you can inactivate the peroxidase because it would give you a unspecific signal too. So you can use some chloride or some different things to remove that. You can also remove, for instance, chlorophyll from the cells, just washing them with alcohol, the filter. So depending on what you need and what kind of samples do you have and what do you want to see, you have to do some previous preparation of your samples. Then you can cut your filter in different sections, and then hybridization begins, then you have these molecular proofs. These molecular proofs have an enzyme attached, the peroxidase, okay? Not the fluorochrome, but an enzyme. This is the difference with the fish, the normal fish, okay? That in each proof you have one fluorochrome. And in this case, we have an enzyme. So we carry out the hybridization, and then you have to choose different amounts of formamide and temperature to do this hybridization, because you want that this proof attached to the ribosomes of your target organism. Depending on this concentration and the temperature, the union will be more specific or less specific. So you have to find the perfect conditions, and you have to do some tests to ensure that you are highly specific, but not too much, okay? Again, if it's published and the proofs are already tested, you will find these conditions. Otherwise, you have to test it. Once you have this hybridization step done, well, there are some washing steps to remove the excess of the proof. And then you have all your ribosomes of your target organisms with the proof attached with the peroxidase. Okay, that's the washing. And then we do the amplification. Now we add the fluorochrome in the sample. Okay, there are different fluorochromes available with different colors. And again, depending on the stainings that you are doing, double or triple stainings, depending if you have chlorophyll in your samples, you can choose a red fluorochrome, a green one, a blue one, whatever. Okay, and then there's the amplification reaction. Then what all the fluorochromes go through these enzymes from the proofs, and this signal, they change the conformation of the fluorochromes. They become right, and they are attached to the membrane of the cell. Okay, again, for fish, you have one proof, one fluorochrome, and that is your signal. In this case, we are amplifying the signal, and this signal goes to the membrane of the cell. So you have much brighter images of the positive cells. Okay. And once done, then you have some washings again, and then you can prepare your slides with all your filters. Here you can do your double stainings with TAPI, with alcohol, with whatever you need. And then you can directly observe these filters in the fluorescence microscope. Then have some examples here for the different applications. Why do we want to do that? Okay, so maybe we want to study the life cycle development of our organism, our culture. There are some steps that are really difficult to see in the light microscope. Then we do the hybridization. This is the dinoflagellate. In red, we see the chloroplast. In blue, we see the DNA, the nucleus. And in green, we have the molecular proof. We can see here from the early infection, the trophon is growing. This image is in 3D because it was done with a confocal microscope. We can also reconstruct.
Вие работите во чачак, така?
Да, јас мислам.
We can reconstruct the situation inside the cell where the parasite is located. Okay, and here we can follow all the life cycle thanks to the amplification and the hybridization of the proof. This is exactly the same for Amebopria. In this case, the genetic material is in red, and in green again.
Да.
Maybe we need to quantify. If we want to quantify our target organism, our target group, we can also do this fish. In this case, they used the proof for chytrids to stain the zoospores, and in this way, determine the zoospores in the sample that belong to chytrids and those that didn't belong to chytrids. Okay, and then in that way, they could quantify the amount of chytrid zoospores that we have in the environment. And other applications. Sometimes you don't know the organism and you just obtain some molecular information of a molecular group that could be of interest, but you don't know nothing absolutely about them. You design a group that target that molecular clade, and then you have your filters, you use the proofs, and you try to find the organism that correspond to that molecular group, okay? In that case, they realized that, again, this was an organism, or this group contained organisms that were parasites of diatom chains. Here they have the images. So that's it. That's the general overview of the technique. You will have the protocol, and you can have a look at it. And just a quick overview of why do we need or why do we use this technique for when studying the parasites.